Hello, and welcome back to What is a Biology? I'm your host, Mr. Johnny Hopkins, and today we're going to be starting a new little small unit called genetics. Just like the basic vocabulary and what you need to know to just kind of go forward with what we're going to talk about in the rest of What is a Biology. So, first of all, we can't really talk about genetics without talking about what an allele is, and that's just kind of a variant of a gene. So, what is a gene? If you recall, a gene is essentially just a single unit that is transcribed on the DNA. And there's a lot of different anatomy to this gene, but we're going to get that later on in the next unit. So when you have an allele, you have a lot of the alleles in the body. So you have 23 chromosomes, 23 in me, but there's also two sets of them, so 46. But, so you have a full set of these alleles. There's a lot of different alleles on the chromosomes themselves. And so we call all of these alleles together a genotype. It's just a set of alleles for a specific organism. And then when you take it and when we actually look at a human being, you look at the phenotype, the physical traits associated with the genotype. So I am a 6'2" human being, and that is part of my phenotype, whereas somebody who is 5'4" has a completely different physical trait corresponding to their phenotype, which the phenotype corresponds to the genotype. So it's essentially what you can see versus what's underneath. And then you have the wild type. And the wild type are also known as, usually shown as WT, is the standard genotype. So when somebody's like, we just looked at the wild type versus this mutants, it's the normal, active, general one that you're going to find in a human body or regular bacteria, and then you mutate it and you see what happens because of that mutation. So, now just a mutation. So we got a deviation from this wild type. And these mutants have a lot. What even is a mutant? A mutant is essentially just one that has a change in the genetic code. So we got a mutation, and what a mutation does essentially just alters the genetic code. So the most common type is a point mutation, where you essentially have a single residue on the DNA, a single nucleotide, and then you change it from like guanine to cytosine, adenine, or thymine, and that changes the genetic code. And there are quite a few different consequences that this can have, and one of them being a silent point mutation. So essentially, you change this G to a C, and nothing really happens, because when you correspond it to the protein that goes down the line, that actually doesn't change the codon itself. It's just a silent point mutation. Nothing actually changes in the genotype or the wild, but you still get the wild type protein structure. Then you have a missense mutation. So essentially the opposite of a silent point mutation, where you substitute a different codon. So instead of having the same amino acid be translated, you have a completely different one. So instead of having alanine, you now have arginine. Ah, yikes, that's not good. And then you have a nonsense, which is a specific type of missense, which essentially has a premature stop codon. So instead of continuing on and making a full-length protein, you truncate it short and you get some bad boy, some bad, unfortunate thing. One thing also that can happen is a frame shift, where the ribosome keeps reading, but then it just kind of happens like, doesn't go forward 3, only goes forward 2, or it backtracks 1 or something, and then you have a completely change in the reading frame, such that you're no longer in the same reading frame, such that, like, say A, T, C, C, G, G, A, C, where we read 3 and 3 and 3. That's how it's supposed to go. But instead, when it reads this guy, the ribosome then goes and just reads this instead. Yeah, whatever. And that just kind of is bad. And like you might get a premature stop codon, you might get a stop codon way too far, you might have a really weird mutant. And so, what do these mutants have happen? Is you can have nothing happen. You can have a silent point mutation, or you can have a missense mutation, where essentially you just have a protein that goes from We have like a mutant that goes from, like, say, alanine to valine. Not a lot of changes there when you go from alanine to valine, because it's pretty much the same structure. And so then you get pretty much no change in function. But you can have a loss in function or a gain in function. So essentially, if we change arginine to alanine, we might get a loss of function where the function of the protein can no longer occur because this single residue, this single amino acid, is critical for the function of the protein. And then you also have a gain of function where you can mutate, say, alanine to arginine, and then you have, instead of the protein binding to just, say, Sonic Hedgehog, it also now binds to some sort of the actin filament. So it has a gain of function, even though it doesn't have that function in the standard biotype. These guys can be very important for different diseases in and of themselves. And so now we're going to get into a little bit more broad topic, so I just talked about how 23andMe, we have 2 sets of 23 chromosomes, one from our father, one from our mother. And that's what this thing is called, a diploid cell. So essentially we have 2 sets of the same genes, you have a diploid. Whereas if you just have 1 set, it's a haploid cell. And so most multicellular organisms exist in a diploid state, but you can have usually lower eukaryotes and bacteria are haploid. So, but we might remember is that not all of our cells in our body are the exact same. We have, autosomal cells, which are non-reproductive cells, and then have the gametes, the sperm and the egg cells. And those guys are haploid. So we have, and what happens is you go from meiosis, where you go from a diploid to 4 haploid cells. Also another thing is that you have yeast. And yeast are a lower eukaryote, and they happen to exist in both the diploid and haploid state. There are 2 different sets. You've got the A and the alpha. And we'll talk about that a little bit more. And one thing that you can actually do is you can take a diploid cell of a yeast and then you starve it. You do starvation, and then it'll actually go into 4 haploid cells. And so that little thing, that little niche of these yeast allows us to study them better. So we have heterozygous. So now we have heterozygous and homozygous. So this is kind of just a hodgepodge of different terms and stuff, which is just kind of how this genetic unit will be. And so what you probably know this already is, since we have diploid cells, we have 2 different types of cells. You can have a recessive gene or a dominant gene. And so when you have a dominant gene, the dominant one takes effect and you only see the dominant occurrence. And then when you have both recessive, then you see a possible, you just see the recessive trait in and of itself. And so, if you remember the Punnett squares, where you just kind of mix and match, that means you have a 25% chance. If you mix 2 heterozygous for a single trait, you have a 25% chance for a homozygous dominant, a 50% chance for heterozygous, and then 25% chance for homozygous recessive. And you have a bunch of different possibilities for these sorts of things. And so now when we go back to yeast, there's also sorts of genes that are haploinsufficient, where you need both alleles, where you need a diploid state, otherwise the cell will die, period. And now we're going into some more different mutants that we have, which are dominant negative mutants. And these dominant negative mutants are where you have where you can have essentially the dominant gene be the gene that is horrible mutation, makes you die, which is which, because it makes you die, usually doesn't show up until later in life, like Huntington's disease. And so the dominant allele performs negative action, whereas typically it's shown that the recessive allele performs this negative action that makes the individual cease, not have normal function, sometimes cease to exist. Okay, continuing on there, now we have something called homolog. A homolog is just an analogous protein, so with analogous being in the sequence structure or the function. And essentially it's just like taking the idea of a protein in bacteria, just kind of crawls from, goes into the transmembrane, goes into the membrane and senses if there's glucose around. And then you got another protein in humans that does the exact same thing, goes to the surface and says, is there glucose here? And that is essentially just a homolog. It's an analogous protein, so they typically have a similar sequence, similar structure, and a similar function. And that is what a homologous protein is. And so therefore, kind of a homologous gene is the same exact thing because a gene encodes a protein. So going back to mutations, you have something called conditional mutants, where this is actually very useful for studying the effects of genes and what the functions are, is that you have essentially a temperature-sensitive mutant, where if you increase the temperature of the medium that the cell is in, then that gene is no longer transcribed and translated, so you no longer see the effects of that gene, so you can see what happens when that gene no longer exists in the cell by inactivating the said gene. And so one of the mainstays of these genetic studies was H. Moeller, who studied Drosophila. Drosophila are just fruit flies. And one of many findings he had was that inbreeding is that usually you find, when you have inbreeding, you can find homozygous recessive lethal mutants. Essentially, when you have inbreeding, you can find some really strange diseases that would not happen if you just, if people from different families of not the same genetic origin were to mate. So that's kind of why it's frowned upon to inbreed between us humans. It's sort of a taboo, if you will. So, another type of mutant is a double mutant. So, essentially, where you have two mutations on the same gene, and when that translates to a protein, you get a protein that has two different functions. It represses one part of its function and promotes the other part of its function. So that can actually allow us to study different signaling pathways for different ways. So essentially, it will bind two proteins. It will bind protein B and protein C, but in this case, it will no longer bind protein B, but it will really bind protein C. So that's something that like a double mutant will occur. And then you have something called genetic suppression, where essentially in a base case you have A and B, they'll associate. But when you have a mutation, the genetic suppression, A and B no longer associate. So this sort of suppression means that the signaling pathway, you no longer can, the effect of the gene effect of mutation on A affects the signaling pathway and everything for B because it no longer can go on the same pathway. Finally, we have something called synthetic lethality, where essentially if you have a lot of proteins in a little bit of pathway or a complex or something, some major thing. And then what happens is if you just mutate some residue in one of those proteins or one of those genes, nothing happens, no matter what thing you pick. But when you pick just 3 of them or more of them, then you just see the cell die. So when you have multiple mutants, then you have a bunch of problems occur and everything kind of just goes bad. So that's the basics of genetic vocabulary. You got alleles, and you can mutate them. You got genotypes that are the set of alleles, and the phenotypes that represent the physical traits of those genotypes. You got a wild type, which is the general one. You can have loss of function and gain of function mutations. You have haploid versus diploid cells, where diploid is 2 times. You have heterozygous versus homozygous, heterozygous, homozygous, homozygous. And haploinsufficient stuff. You got dominant negative, which are like Huntington's disease, which is very bad. Homologous, which means cross species, similar genes, proteins. Inbreeding is bad. Double mutants, which means that you can have repressing and promoting effects. Genetic suppression, where you no longer bind to a certain protein in the pathway, so the pathway just kind of collapses. And synthetic lethality, where you need to have multiple mutants in order to see an actual effect on the cell. So that has been What is a Biology? The basics of genetic vocabulary. I'm Mr. Johnny Hopkins. Thank you.